lecture is postponed, so we have a lot of time.
Okay. So now you can see the screen.
Yes.
So I was just explaining about the different types of stem cells, the categories. So now I will go how exactly we will study this. So pluripotent stem cells. So the question comes, how do you establish? What are the prerequisites? So obviously, since it is embryonic stem cells or pluripotent stem cells, so first thing, inherent pluripotency, as I mentioned, that it will come from the embryo, developing embryo. So the embryo can be either, it can be obtained from the in vitro fertilized embryo or somatic cell nuclear transfer. I will discuss about this also. Even in the animal husbandry, the parthenogenetic embryos, one can get this. Also, in addition to that, one can get the pluripotent stem cells from somatic cells by using reprogramming, where one can think of using the embryonic specific genes which will make the somatic cell behave like embryonic stem cells. So I'll go one by one. So from the developing embryo, when the particular stage, like as we know that in inner cell mass cells which can give rise to the embryonic stem cells. So the developing embryo, when we take, we can either do the direct embryo culture, then based on the conditions that we are providing, so the inner cell mass cells can be obtained and those can be further propagated and grown to make the embryonic stem cells. Or one can think of using laser microdissection, where one can retrieve the inner cell mass cells and culture. The third approach is immunosurgery, which is very conventionally used for establishing embryonic stem cells. I will show this also how it is done. And next, as I mentioned about somatic cell reprogramming, I'll discuss about that too. So first, coming to the somatic cell, if you have to do, or the embryo you have to take. So one is direct embryo culture, and the second immunosurgery I mentioned, or the laser microdissection. But for getting embryo, either you are taking from the in vitro fertilized, where artificially you are doing those couples who go for the infertility treatment. So the embryos which are discarded, not implanted into the uterus, those embryo usually we collect the discarded embryo, and that we take for further embryonic stem cell isolation. The other technology is called somatic cell nuclear transfer, where a somatic cell itself, you can convert it to the embryonic like stem cells. So how it is done, as the name suggests, somatic cell nuclear transfer. That means any cells you are taking, for example, any patient, any somatic cell in our body, skin cells. So you take the cells, so this will become the person or patient specific. And you take one egg cell or oocyte, remove the egg nucleus, because nucleus is the one which engrams the genetic material that provides us our own identity. So if we remove the nuclear material, that means it has only the cytoplast or the cytoplasm, all the thing, but except the nuclear material. So this enucleated egg you take, and then any cell, the somatic cell will have the nuclear material, which is our own identity, that you fuse with the enucleated egg. And this one is stimulated further to bring to a stage of blastocyst, where the inner cell mass cells can be retrieved, cultured, then you can get the embryonic stem cell like pluripotent stem cells. In animal system, what happens? This entire, when it comes to blastocyst, it is implanted into any surrogate mother. Then you are getting the pups. So that is called reproductive cloning. But when you are stopping at this stage and taking the inner cell mass and culturing those further, that is called therapeutic cloning, because you are not implanting it further, neither you are getting, especially in human case, because of the ethical concerns, we never implant the somatic cell like SCNT mediated embryo developed. So those are restricted or the stopped at this stage, and the inner cell mass cells can be taken and cultured. So these are the steps here. It is mentioned on the left side: enucleation of eggs, insertion of somatic cells, activation of reconstructed egg, cleavage of the reconstructed egg so that you will get the stage of this blastocyst. Then you can culture those and then ESL line derivation can be done. So as we all know about Dolly is the best example of somatic cell nucleotransfer. In fact, this was done as early as 1952 and 1962. John Gordon, who also shared the Nobel Prize with Yamanaka in 2012. So there they have done actually in embryo, the experiments are done. So Briggs and King, they did very early stage embryo using enucleated egg. They have taken from the very early stage embryo, they have taken the nuclear fused and most of them developed into tadpoles. But the same experiment when by John Gordon, it was taken from fully differentiated intestinal cell. So that using the same strategy, taking the enucleated egg and fusing that, less than 2% develop into tadpole. What does it mean? That means stage of the cell that does matter. So if you are taking very early stage, that means they probably retain much more higher capability of developing to embryo. So the stemness of the cells are much higher compared to the cells which are fully differentiated or mature cells. The same strategy as I was telling, using that Dolly has been developed and now many more organisms in fact have been developed using the SCNT technology. But this is not free from any limitations either. First of all, if you think the availability of oocyte, because you need to enucleate, so availability of oocyte is one of the limitations. Second is heteroplasmy. As I said, you are removing the nuclear material, but the cytoplasm, the mitochondria and DNA, everything is there. That's the reason why you are getting heteroplasmy. Sometimes even the epigenetic signature maintenance, it is another problem because during development, we have got I don't know how many of you are familiar with epigenetics. It's not only the genetic material that determines or dictates what we are, our physiology is affected, different activities are affected due to gene expression. Even the epigenome, the epigenetic modulators, which is the environment, that is also playing a role. So the way DNA is modified or when it has to be expressed, when it should not be expressed. So the epigenetic signature, that also sometimes it is methylated. Then de novo methylation is taking place, demethylation is taking place. Those are the epigenetic signature also should be maintained during development. But using SCNT, since we are somehow not following the regular developmental pattern, we are altering that. Sometimes even the epigenetic signature maintenance, there will be some limitations since. And as I also mentioned, the differentiation state or the stage of the cell that matters. So the earlier the cells, like what we have seen the earlier experiment, Briggs and King versus John Gordon. So the differentiation stage also influences the success of this. And sometimes even the memory from previous gene expression, that means incomplete reprogramming happens. For example, if it is taken from muscle cells, so after SCNT, when the ICM cells are taken and embryonic stem cells are established, when they were differentiating, they are directed more towards giving rise to muscle cells. So this is the previous gene expression memory is written, that is one of the limitations. So to rule out this, now people are thinking even using rabbit oocytes, whether we can substitute, especially in case of human, it is not permitted to bring to the stage of the child development or implantation, but we can stop at the stage of blastocyst and take the inner cell mass cells. But for that, whether one can utilize rabbit oocytes, that kind of research also is going on. So Dolly, I already gave the example, I'll just skip this. Next comes parthenogenesis. That is also in the animal husbandry. There they utilize super hot development. So there, this is a kind of vegetative propagation. So you are taking the oocyte, then you are trying to trigger by artificial stimulation, you tweak or the trick that oocyte thinking that it has been fertilized, then bring to the stage of blastocyst. So at this stage, so this N becomes 2N, so you are diploidizing that, and then this blastocyst inner cell mass are taken, and you can also make the embryonic stem cell line establishment, or you can implant this, which is happening in the animal, actually many of the veterinary institutes. So if you have any specific hot where it is much more superior, So by using the parthenogenetic means, they are developing the organism, which is not ethically permissible in case of human. Then comes the regular embryo, which I was telling about the different techniques that are followed to take the inner cell mass. So by SCNT means when you are bringing to ICM stage, or by parthenogenetic means also you are bringing to ICM stage, or you are taking from the IVF or direct embryo culture. So there at the blastocyst stage, if you see, this is the outer covering, which is the zona, and this is the inner cell mass. Zona is the mucilaginous covering. So using specific protease, we digest the zona. Then only you are getting this particular structure, outer covering of the trophoblast and inner cell mass. Then here the immunosurgery that I was telling, what happens? We use specific antibodies. And since we are not permeabilizing, the antibodies will attach to the outermost cells. Then when you use complement against that, complement function is to chew wherever the antibody is bound. So only the outermost cells where the antibody is bound, those cells will be disintegrated and you will get only the inner cell mass. This is in fact we have done in our lab. That is what I'm showing there. And using the same, so this is from the human embryo, discarded embryo. Human embryonic stem cell line has been established, and mouse, these are commercially available. Even human is also commercially available. This is showing the mouse embryonic stem cells. This is human embryonic stem cells. So mouse stem cell, if you see, this is actually in background, you can see some cells which are fibroblast. So they form like a feeder layer, or they provide specific factors for the cells to remain unspecialized. And those who are working on embryonic stem cells, it's very critical to keep in mind that the cell should remain unspecialized or in a blank slate-like state. Since these cells are pluripotent, so unless and until we maintain these cells in unspecialized state, we cannot direct them to cells of our interest. They will lose their pluripotent nature. So we try to manipulate the culture conditions and maintain the cells in unspecialized or undifferentiated state. So either it can be mouse or human or any species, one can actually derive the embryonic stem cells from. And these cells, further it can be, as you are interested in directing those to specific lineage, one can also play with the conditions and direct those to differentiate. Next comes whatever we saw now, the inherent pluripotent cells. Now come the induced pluripotent cells, which are reprogrammed. So here the beauty is what happens in embryonic stem cells. As I told, this is coming from the developing embryo, and these have enormous potential to divide. Indefinite self-renewal they carry. At the same time, they can give rise to all the cell types of our body. But if you are thinking in terms of therapy, so there, if you have to have patient-specific cell line development. So since embryonic stem cells are coming from a different embryo, those cannot be similar to the patient or whichever person you are requiring to imply these stem cells for. So in that case, for the immunocompatibility or no immunological rejection to take place, one has to develop patient-specific cell lines. Adult stem cells, they also divide and differentiate, but as I told, those are multipotent or unipotent, depending on which tissue they are derived from. So they are limited or restricted in their differentiation potential. So in that context, what happens? Because they are limited in cell division or proliferation or self-renewal rather, and differentiation. So the cell number that one may require for cell replacement therapy or in clinical setting might be limiting. Whereas embryonic stem cell having the property of indefinite self-renewal and directing those to any cells of your interest, so the cell number may not be a problem there. But problem there is it is not compatible to the patient. You have to have patient specific. So combining the beneficial effect of the two, Yamanaka's group, what they did, they tried to reprogram or rewire the system. They took any cell, like I can take my skin cell and then develop into my, like a Nivedita specific embryonic stem cell or induced pluripotent stem cell. I will treat the system, rewire the system using specific factors then it will resemble my own self. So in that case, it will have the patient-specific cells. So what they have done basically, they have screened 24 different factors. From that, they have nailed down to 4 such factors, which is called KSOM, KLA for SOX2, OCT4 and CMYC. Those are also regarded as Yamanaka factors. Subsequently, there are other factors which also have come into being. Even using a single factor, one can trigger the somatic cells to behave like embryonic stem cells, where actually you are bringing back to the naive state. That is called differentiation. So your differentiation from the normal cells, when it matures, it is differentiation. But when you are reversing the process, it is de-differentiation. So somatic cells using specific, and what is the significance of these markers? These are highly expressed in embryonic stem cells. That's why using these, so you are reprogramming the somatic cells to behave like pluripotent stem cells. That's why it is called induced pluripotent stem cells. The other advantage of these, not only it is patient specific, which will be non-immunogenic. If you have any genetic disorder, the same gene signature is written in this, because it's the nuclear material is coming from the same patient, cell is coming from the patient, it is patient specific, and the nuclear material being that the same defect also is recapitulated. That's why you are getting a disease in a dish. So that is another advantage as basic biologists we always look for. See that whether we can replicate, because many times we use animal models to understand the disease process. But if you can mimic the same disease process in your dish using the iPSCs strategy, so there you have the disease in front of you, then you can see what might have triggered the disease and what all modulations or modification we can do so that we can reverse the disease process. Now the era of even gene editing is coming. So if there is any genetic defect, one can probably reverse the process. So this is the advantage of induced pluripotent stem cells, which is combining the beneficial effect of both embryonic stem cells and adult stem cells and bringing the patient-specific cells into picture. I hope I am clear to all of you. If any of you have any difficulty on understanding, you can interrupt me. I'll be happy to answer. Then this is showing the example we have established also from the skin cells. After a while, when you are putting the Yamanaka factors and they form from the skin cell, they get converted into pluripotent stem-like cells, which I showed also embryonic stem cell. They form colonies, and these are the unspecialized or undifferentiated mass of cells. Those are further propagated in culture, frozen down for the stock maintenance, and we try to direct those to specific lineage for differences. Now coming to adult stem cell, I'll show you another video here for adult stem cells.
After the embryo has grown into an adult, many of the body's cells continually die off and need to be replaced. For example, millions of red blood cells need to be replaced each day. This is the work of stem cells known as hematopoietic stem cells, which are located in the bone marrow. Residing in a complex cellular environment, the hematopoietic stem cell is able to generate precursors to every type of blood cell. And indeed, these stem cells have been used by physicians for decades to replenish patients' blood supplies following radiation treatment for cancer. The stepwise differentiation of a stem cell's progeny depends on combinations of genetic and environmental factors that gradually steer these cells into forming a specific lineage. This process involves different sets of stimulating factors operating in the cell's environment and its surface membrane and within its nucleus. Each step down the path draws it closer to becoming a functional red blood cell and entering the bloodstream. This following testing turns over even faster than the bone. At the microscopic level, this epithelial surface consists of billions of projections called villi, with stem cell containing areas known as crypts and basins. Contains smaller cloned stem cells that maintain their own numbers while also producing progenitors. Cells came out shock from the tips of the bone.
And I think all of you must have seen also the lizard, the tail. It sets up and it grows again. So that is the potential of regeneration. So the stem cells, in fact, are playing a role. Even the birds we see, the flight muscles, there is also a lot of regeneration is happening. In our body itself, liver, if you just take a portion of liver, it regrows. So that's why the regeneration potential is there in adult stem cells. They have, because of the potential to divide and differentiate, they can regenerate, but not all the cells. But in fact, in our adult body, in discrete pockets, as I was mentioning, in specific organs, these stem cells are localized. As when it is required, many in a quiescent state. So those are physiologically active cells, but they are not dividing. So that particular niche or the microenvironment helps these cells to remain as it is, as stem cells or quiescent population. And these stem cells, and when there is some insult or some trigger is given, then they again come back to the mitotic state. They divide limited number of times and differentiate. But when we see the disease process, so what must be happening there? Because of their limited self-renewal capability and differentiation ability, so the cells, depending on the trigger or the insult that is there in the surrounding, so to some extent they can retaliate that process and they can give rise to replenish the cells. But once the threshold is gone, you are seeing the disease process. So as it is shown also in different organs, we have stem cells. So here also the question comes, how will you get this? So you have to think about the tissue source. How do you isolate or retrieve? Whether you will utilize enzymatic digestion, because these are tissue cells. So you have to get single cells, or you can think about doing explant culture. Then you have to get the cells, maintain, differentiate. And another characteristics of adult stem cell, that it's called, those are highly plastic. So one, I was telling about differentiation, then de-differentiation when you are inducing pluripotency. The other is called transdifferentiation. So trans means it is actually crossing the lineage barrier. One cell is becoming another cell type. The muscle cells which are destined to become muscle, if they become nerve cells, then that is called transdifferentiation, or vice versa. So that potential also people have studied. And there are still controversies in this regard, but some cases it is, in fact, it has been shown also in case of animal studies. Some groups, they agree that it is actually transdifferentiation phenomenon. Some think it might be some paracrine influence, but I will not go that much deeper into that, but just understand the difference between differentiation, de-differentiation and transdifferentiation. So the plasticity of cells actually is materialized through this transdifferentiation process. Then once you get these cells, one has to characterize, enrich, purify, and do whatever the downstream one experimental plan or somebody has or implications have in mind, those only we have to do. I'll show you some examples here. So these are the mesenchymal stem cells. Our blood has two types of stem cells. One is hematopoietic stem cells, one is mesenchymal stem cells. Mesenchymal stem cells, in fact, support also hematopoietic stem cells. So these are multipotent stem cells which can give rise to bone, that is osteocytes, fat, adipocytes, and cartilage, chondrocytes. 3 different cell types can be formed from this. So the examples you are seeing, this is from the bone marrow. This is from umbilical cord blood, which are discarded, umbilical cord, placenta. So the cell morphology also, those are basically the fibroblast-like morphology or spindle shaped, but some, depending on the source also, the cells have different self-renewal ability, as well as the morphology also changes, varies a bit. So these cells further, once you get that, then we have to think how to characterize. So be it embryonic stem cells or adult stem cells. So now we know how to retrieve the cells either from developing embryo or from adult. Then once you get that, one has to understand the specific characteristics. How will one know that I am Nivedita? Because I possess specific characteristics. Same way the cell, once we get the cells, they have to have their own identity. For that, we need to see, and the stem cells basic characteristics, one is the unique features are self-renewal and differentiation. The self-renewal in embryonic stem cells, it's indefinite self-renewal, which is nothing but renewing themselves. Because they possess very, very high telomerase activity, they can divide or self-renew themselves indefinitely. In contrast to the adult stem cells, where the telomerase activity is less, that's why they have limited self-renewal ability. And differentiation, as I have already mentioned, those can be either pluripotent, multipotent, unipotent, and those can also transdifferentiate. Especially in case of adult cells, we see transdifferentiation. They are highly, and they do possess regeneration potential, as I gave the example of liver. So that is a mean of repair in adult body happens. And these cells are amenable to genetic manipulation. In fact, the recent very excuse me.
Sorry.
Okay. So the recent, very recently, last week actually one paper has come. I will show you that reference at the end. So in Nature, one group from China, they have published whether we can, like the way somatic cells were converted to pluripotent cells, like our normal mature cells can be converted to pluripotent embryonic stem cell like cells, whether embryonic stem cell, the pluripotent one can be again brought back to the much more naive state like zygote, totipotent cells. Small molecules, they have succeeded in converting the mouse pluripotent stem cells into the totipotent like cells. Because the technology, that's why the newer technological advancement. Here you have to understand the basis. What is the rationale behind? Like somatic cell, if you are incorporating specific genes which are expressed in embryonic stem cells and they are getting converted to embryonic stem cells, the epigenetic machinery also is rewired. Genetic machinery is rewired. Same way, pluripotent cells converting those to totipotent, again the rewiring or reprogramming of the system is taking place. Same way, direct reprogramming also is possible. That is also, in other words, can also be called a transdifferentiation. One cell is becoming another cell type. So, Dr. Vijay Tiwari, he was there last week. Their group, they have shown if a specific transcription factor, if you are trying to incorporate in any cells like NeuroD1, you can convert the cell because NeuroD1 is expressed in neural stem cells or neural progenitors. So you can convert any cell to neural lineage by triggering the system or activating the machinery using specific factor. The same way they have converted the pluripotent cells to totipotent cells. So with these characteristics, how will one study self-renewal? As I mentioned, it can be indefinite or restricted. So symmetric cell division is associated with indefinite self-renewal. One stem cell gives rise to 2, then 2 daughter cells again give rise to 4, and so on and so forth. It goes on. So embryonic stem cell, because of the high telomerase activity, they can undergo symmetric cell division and give rise to indefinite number of cells. Whereas adult stem cell, depending on their capability, they can give rise to both symmetric, they can undergo symmetric asymmetric division or entirely asymmetric. In this case, the stem cell, adult stem cell can give rise to 2 stem cells, then one will give rise to 1 stem cell and 1 progenitor or precursor. Same also true in the other case. And asymmetric one stem cell will give rise to 1 stem cell and 1 progenitor. The progenitor can further divide to more mature cells or 2 progenitors, and this one stem cell it will retain and 1 will become progenitor. So this is the way the self-renewal or cell division takes place depending on the stem cell in question. And after certain time, the mature cells, especially because every cell cycle we know the telomere shortening is happening, so they will come to a stage where replicative senescence will happen. No further division is possible. But because of the high telomerase activity, stem cells can self-renew. Then comes the differentiation. So once you have the characteristics, you have studied the self-renewal, then comes to differentiation. So you have to tweak the system, manipulate the conditions like the way we require specific factors for our growth, protein, carbohydrate, fat, different growth factors. Same way we provide in the culture condition specific factors to direct the cells to specific lineage that we are interested in. Even cell-cell adhesion, the junction proteins, the extracellular matrix component, those also play a major role. How to direct or dictate the cells to become one cell type versus by inserting or deleting, or now the gene editing approach also, one can convert one cell to another, as I was telling some time back about rewiring the system. Same way one can think of utilizing different strategies and how to modulate the entire program so that one can convert one cell type to another cell type, be it differentiation or transdifferentiation, de-differentiation, etc. So this, what I said, it's not a very like a haphazard kind of system, very programmed way it goes. Like the way when we go to any, like a traffic signal we encounter, there we see three different lights. Right? One is red, yellow, and green. Green gives the green signal, yellow it's a warning that it is coming either to halt or you have to go proceed. Red light stops you. The same way cellular machinery also has got the same kind of machinery which is very defined and programmed way it proceeds. And if it doesn't happen that way, then all kind of diseases we do see. So when the system is very well planned and differentiation is taking place, the development is proceeding, normal development without any defect, the baby is born, and we are having every process in a normal way, like what happens in highways. But when you come to a station or airport, everybody is in a hurry. Same way, if in our system, if some kind of insult is there, so they are not following a typical pattern, you see this kind of chaotic situation, sometimes it leads to fatality. Even some cells die, there will be also embryonic death, even the cancer that we are seeing, because in normal scenario, what happens, the cell divides and differentiates into mature cells. But if some kind of insult or something, the microenvironment or depending on our lifestyle, because now cancer is considered a lifestyle disease, same also is diabetes. So in this case, what happens, the normal scenario or normal development is not proceeding. So here some cells, they get mutated or the environment plays a role, then the specific pattern is changed. Alteration in that particular pattern, it causes specific diseases, and then we see actually the triggering of cancer or any other diseases. So one has to think that way, how to treat. If you understand this particular profile or the developmental hierarchy, then we can see what is the trigger which is causing the disease process, as I was stating you earlier. So I'll show you some examples. So from embryonic stem cells, we have also directed those cells to differentiate into neural cells. As you can see the processes, the neural processes where the axons and dendrils are seen, and neurotransmission also we have shown also in picture, and this is actually showing a specific signature molecule. By labeling that, we have known the identity of these cells as neurons. Same also is true. Not only seeing this identity, we can also study live how the cells are differentiating or dividing, what cells are giving, how they are interacting. Actually, in a culture dish under microscope, when you observe, they look like very static. But when you do live cell imaging, you see how dynamic these cells, especially you see, actually these are labeled with GAP. So you see they not only move, they talk to each other. Through that only the neurotransmission is provided, or it goes from one to another, and this interaction. So when cells are brought together, because of the cell-cell interaction, the intercellular communication plays a major role in triggering the system and then defining specific cell state. So the cells are specialized or they become one cell versus other. The same thing happens when we are taking from the developing embryo. So 3 germ layers are developed. Depending on where the cells are located, what kind of morphogen gradient is present, the specific cell types are defined. Same in vitro, we try to manipulate, provide the condition, mimic the condition, and direct the cells to become one cell versus other. So that was ectoderm derivative, the neural cells. Here you see the mesodermal derivative, the cardiomyocytes, which can see the spontaneous pulsating activity in these cells. They also show specific wave-like pattern. So this shows actually these are physiologically active cells. Whatever we are generating in vitro are also doing their functions as it happens in our body. Then next comes the skeletal muscles. This is actually a slide borrowed from Duke University, and this is from human embryonic stem cells. We have differentiated into skeletal muscles. So this is demarcated by specific molecule, that is skeletal myocyte specific genes. So we have shown that these are skeletal muscles. So these are mesoderm derivatives. The next comes the endoderm derivatives, pancreatic beta-like cells, which are the ones actually we know also in diabetes, depending on what kind of diabetes, these cells don't function. In fact, the function of these cells, glucose induced insulin release, they do show. So what basically it shows, when you are having embryonic stem cells, you can direct the cells to all three germline derivatives, be it ectoderm derivatives, specific neurons, different subtypes of neurons, dopaminergic, serotonergic, astrocytes, oligodendrocytes, these are the myelinating cells which protects the neurons, or mesoderm derivatives, skeletal muscles, cardiomyocytes or cardiac muscles, even smooth muscles, then endoderm derivative beta-like cells. So this shows the embryonic stem cell pluripotency. Once you understand how the cells are differentiating, once you differentiate, then we see what are the factors underlying this machinery, how the unspecialized cells are becoming specific, one cell versus the other. That is what the research that goes on in our lab, to understand how the cell decides to become one cell versus other, because initially they are just ball of cells. Then what triggers them to become nerve cells or cardiomyocytes or pancreatic beta cells? What are the key genetic component, the machinery available there, the epigenetic machinery, and how we can trigger the system or direct the system so that we can get the cells of our interest abundantly, which can have possible downstream implication in cell replacement therapy. Same also true in case of adult stem cell. The example I am showing, the mesenchymal stem cells, what I showed earlier, those have been differentiated into osteocytes, the bone cells, adipocytes, the fat cells, you can see the lipid vesicles, also chondrocytes, the picture is not here. And not only that, the plasticity, as I was telling, they can trans-differentiate, like the nerve cells can give rise to blood cells and vice versa. Same also we have shown in the next slide I am showing, the mesenchymal stem cells that you saw, which are showing the spindle-like or fibroblastic morphology, based on the condition that we have provided, those can also differentiate to neural-like cells with processes. As you can see in this video, just note it here, the cell that was there earlier has formed a neuron-like structure. So this is nothing but a trans-differentiation phenomenon. So the mesenchymal stem cells are becoming neural-like cells. Same also is true. The next slide would show you the endothelial differentiation. Here, you can see those are forming vessel-like structures. So the mesenchymal stem cells forming vessel-like structure, which in the static picture shown here. So these are the parameters we study to show or see that these are in fact differentiating and de-differentiating or trans-differentiating into different cell types. Now, I will go to the next, the implications. If anybody is having any queries, now I can answer from the first part. Does anyone have any questions, or I can take the questions at the end if the time permits?
Good afternoon, ma'am.
Yes.
Ma'am, I need to ask you regarding that epigenetic factor. As you recently mentioned that there are some factors that a cell carries a whole genetic material, liver cell, heart cell, each of those cells have same genetic material, and the factors, there are certain factors that differentiate. So, have those factors been identified or specified to which causes that differentiation?
Yes. So you are asking how to identify?
Yes, ma'am.
Okay. That is what actually the research will do. we try to identify the factors. So we now know, even in the lower organisms, even in mouse, and so we know specific morphogenetic gradients are there. So if something is known in one organism, we try to see whether anything similar is present, whichever system we are studying, or there are many different newer technology and strategies are now identified. So if you don't know a specific factor, if you want to see some specific transcriptional, like a specific gene regulatory machinery underlying a specific developmental process, so there what we try to do, we do RNA sequencing, even exome sequencing. Now even small RNA like the epigenetic modulators, if you want to study, so by sequencing, doing sequencing, we try to identify the novel ones. Then we try to see if you find the novel factors, whether those are, because there might be some non-specific things coming also. So in that case, once we identify something, we try to validate. So for validation purpose, as you know, any kind of signaling, because any of the genetic signaling or epigenetic machinery, whatever is there, when you are talking in terms of epigenetic machinery, this can happen at the chromatin level through histone modifications, or even at the DNA methylation, as you all know. So that stage, if anything is playing a role, then we do also sequencing. Bisulfite sequencing will show the CpG island mediated machinery. Even the methylation status, histone methylation status, acetylation status, those one can identify using specific, because there are chip, that chromatin immunoprecipitation you see, even the chromosome, that whatever the promoter, the gene machinery occupancy by specific factors. Then we validate using the specific whatever we have identified, whether those are the ones, either by blocking or overexpressing that particular factor, or looking at the chromosome or the chromatin, the occupancy of that, whether the specific gene is activated. Then once we know, we validate, then try to see whether this is having a specific role or not. Did you get my answer?
Yes, ma'am.
Get the answer? Okay.
Thank you.
Okay. Anybody else?
Ma'am,
Yes.
could you tell me a little about the use of induced pluripotent stem cells to study wound healing? Like whether it is a good model to be used, and whether it is recommended to use it above like other options like ex vivo or animal models.
Okay. So in fact, the implication that I was supposed to go now to talk about the implications. So anyway, so you mentioned about wound healing. So in that case, for example, you will take these blood vessels, right? The skin we have to replenish. So if you are taking the iPS patient-specific skin cells, then we can get the, even now what I was telling you, the recent this thing, there is another study also they have shown, you can do not complete reprogramming, transient reprogramming. So even the aging research that shows like you can go back by 30 years in your aging process. So what they have done, they have taken from around 28 to 45 years, like a patient group, taken the skin cells, and they have done the induced pluripotent like iPSC, the same Yamanaka factor induction. But instead of waiting for a month or so, by 17 or 18 days, they have taken that and seen that epigenetic signature and all. And they have what they have seen, the DNA methylome profiling and other profiling when they study, they have seen it is resembling much earlier stage by like which was supposed to be 30 years ahead of the aged cells, the pattern, it is coming 30 years behind. So they are even trying to reverse the aging process. The same way when you are talking about wound healing, so you are replenishing with healthier cells. So be it like you can either think of even mesenchymal stem cells, you are converting those like a transcription system is one way, or if you are making the iPSCs or a transient way, you are modifying that and bringing back. So the capability of the cells will be retained. Then you can convert those, and now even the tissue engineering era, where you can design even the artificial skin. that can be replenished. Not only iPSC, you can make even specific scaffold, grow keratinocyte, fibroblast, and make the artificial skin. That can also be replenished. So one can use iPSC or engineering approach. Now with 3D printing, you can print. Pardon me?
Your signal is breaking a little bit.
Okay.
Now it's fine.
But now it's fine?
Yes, ma'am.
But could you hear me?
Yes, ma'am. I was able to hear you until like the last part.
Okay. No, I was telling that whether you want to use iPSCs or tissue engineering in the 3D printing. Now the era of 3D printing, where you can even print. Now artificial skins, in fact, people have done already. And you can even print. So for wound healing, one can think in these directions, basically.
Okay, thank you.
Anybody else has any question? Okay, then I will move to the next part. And the first question, yes, when I was answering, if you have any specific signaling, you can also activate or inhibit the signaling, and then you can also add the factors for specific signaling is underlying specific developmental process or not. So, okay, now I'll go to the implications. So, obvious question comes, why would anybody study stem cells? So, as basic biologist, my interest is to understand the intricacy of development. As I was telling, if I understand the normal development, we can easily address what goes wrong during any disease process. But for applied science, because that is what draws major attention, one can use these stem cells as a diagnostic tool. As I told, by using induced pluripotent stem cells, if you have any genetic defect, one can replicate the disease itself in a dish. So one has the disease model. So one should not go to the animal level. Even many of the diseases, we don't have animal models present. So in that case, iPSC is the best substitute to understand the disease itself. Then this can also serve as the predictive model for drug toxicity assess. And as we know in the veterinary industry, livestock plant improvement, one can also generate those. And therapeutic implication, that is in the medicine field or the clinic, one can get therapeutic antibodies, proteins, even cells for cell replacement. And finally, the cells, tissue or organ for transplantation. So that will have major implications in the clinic. I'll just go one by one. So what way one can think of? It can be either preventive, reparative or regenerative. Regenerative, already I talked about the liver. So liver has the intrinsic regeneration potential. Then if you can somehow to repair the process, prevention is something in our hands. So if you can lead the healthy lifestyle with exercise and all, many of the things can be prevented. But if it is a genetic defect, of course, it is not in our hand, but that we can actually repair using the tools now available. Or if we can think of activating the endogenous machinery, since we have got tissue-specific stem cells, correct the defective gene by gene therapy. Then if cells need to be replenished, so one can generate the cells of interest, so replenish the damaged cells, replacement of defective organs using tissue engineering and organ transplantation. So these are the tools available. First, the gene therapy. So gene therapy is nothing but you are replenishing the defective gene with a healthier counterpart. So how will you deliver? For that you require a vehicle. Usually viral vectors are used. So using the specific whatever gene you want to replenish, you clone that and then give to the patient. So it can be either through stem cells or it can be directly injected. So we know this year itself, there was first pig to human heart transplant was done, where in fact the surgeons in Maryland Medical School, they transplanted pig heart by knocking out 3 pig genes and adding 6 human genes, so that it not having graft rejection. But unfortunately, the patient died after 2 months. But what it shows, the basic technological advancement, which the rationale behind how it is working. So one can probably tweak the system to make it specific to the same, because now organ transplant, getting that on time is a problematic thing. So people are thinking whether you can convert the pig, because we have much more resemblance to pig. So if pig heart can be transplanted or any organ from pig, But there are also some concerns about the Yersinia virus. But that's why the scientists have tried somehow to make it compatible to human, whether you can inactivate or knock down specific genes from the pig and add human specific ones. So in principle, it is possible. Using the same genes have been, Jim Wilson at University of Pennsylvania, he is the pioneer in the gene therapy field. So he has done actually many of the patients, even including ALS, has tried, but in between that got into some kind of setback. But now again, it is actually gaining the momentum. Next comes the cell replacement therapy. So as we know, different stem cells already I have explained. So if you can differentiate those cells much more, and depending on the requirement, so there when you are thinking about stem cell replacement therapy, one has to keep in mind it has to be compatible and how often one has to be given, what stage of the disease it is there, how much damage is there. All those things one has to keep in mind, and which stem cells you have to take. If I have some problem in brain, obviously I will not cut open the brain to get access to the brain stem cells. Majorly which has gone to the clinic is the hematopoietic stem cells, as we know. In leukemia, that is what has gone first to the clinic, and this is well established. Same also one can think from the when we are extracting the tooth, even there also we have got stem cells. Umbilical cord, cord blood, which are the waste products, one can get the stem cells, and skin cells like patient specific ones, one can convert to patient specific cell types. One can get the patient specific cells, and where one can think of avoiding the immunorejection. So this is what I was telling, that this is the most widely used stem cells in clinic for cell replacement therapy, the hematopoietic stem cells. And earlier days also, the aborted fetus, they have utilized for the stem cell therapy. So this is before surgery, where the dopaminergic neurons have been transplanted. This is after surgery. But again, the same question comes from aborted fetus, whether you can get enough cells, and obviously from adult, you cannot probably take these cells, right? You cannot cut upon the brain to access the stem cells and get those. So that is impractical. Even though we have stem cell population, but to access that is a major limitation. So that's the reason why people are thinking how best one can utilize which stem cells, whether one can transdifferentiate, even though it is controversial, iPSC or direct reprogramming one can think of, but still these are at infancy. Lot of groundwork needs to be done before it goes to the clinic. Now in Japan, in fact, they are doing induced pluripotent stem cell therapy for the retinal defect, even Parkinson's patients also. It has got the green signal to go to the phase 2 clinical trial. So this is the study we have done in our lab using mouse embryonic stem cell derived dopaminergic neurons, the green ones. So those have been actually implanted. We have generated the hemi-Parkinsonian animal model. One side is the defective, another is intact. So after implantation, you can see it is restoring or coming back to the level of the normal ones, both the sides. This is the defective side. After implantation, we have seen, and even we have seen the physiological relevance of this, that secreting dopamine. So I will not go much deeper into that. So it is just showing that strategy or the technology available which one can think of utilizing. And this is just a cartoon showing, like if you have to do any transplant, for example, Parkinson's disease, dead nerve cells not producing dopamine, the subventricular region cells. So you have to implant stem cell. In this case, you have to think what stem cells you have to keep in mind. Then those should be physiologically active, producing dopamine. And after implantation, they also should integrate into the system. So this is what the cartoon showing, which stem cells you have to determine, what stem cells you have to take for implantation, then match with the patient, give back at the right place so it should function normally, and integrate. And that is what the cell replacement therapy means. It should function as the endogenous cells that usually function in our body. And therapeutic cloning, as I have already discussed, using the induced pluripotent stem cells, one can think of generating patient-specific cells using SCNT approach, direct reprogramming, or iPSC. Now, which has drawn major attention, one can think of generating the cells of interest, and wherever you have problem, you can replace the cells. So this is another example showing that they have done as well as 2015. Now there are actually many more such activities are going on. As I told also in Japan, they have done already. So this is something iPSC, human iPSC derived glomerular. This is another very difficult cells, in fact, to get, kidney cells. So those after implantation in mouse, so they have shown the integration and they have established all the vasculature. Because once you give to the patients any cells, they have to integrate into our system, and vasculature needs to be established so that the blood flow and every nutrient also has to be there. So the restoration of the endogenous machinery needs to be there. And that's what people are studying in majorly in the animal system first, and then that can be extended or extrapolated for the human system. Now we know the gene-mediated therapy, cell replacement therapy. So since our organs are in 3D, so now even as researchers, we are trying whether we can mimic even the organ in the 3D modeling. So miniaturized organs are called organoids, or you can print the organs or develop bioimplants. So what does it mean? It is the interdisciplinary field that combines, that is what I was telling, the STEM field, engineering and mathematical modeling. That is what comes into picture here in the field of tissue engineering, where you can reconstruct or recreate any kind of tissue organ for the replenishment of the repair or replacement of the organs. So these are the examples I have given here. So for example here, so what are the requisites? So if you have to make the 3D modeling, so one has to have the 3D structure established. For that, you require a scaffold which can be either natural or synthetic. And those need to be biocompatible, non-cytotoxic, histocompatible, immunocompatible, non-mutagenic, and over a period those should be degradable, because we don't want any foreign material to be there in our body. So after the endogenous machinery works and you get the replenishment, then this has to degrade. And the other cellular component is stem cell. So you have the scaffold, so you have the cells, you try to put together, for example, the donor tissue here, the cartilage. The knee replacement, as we know, the osteoarthritis. So for cartilage, for example, so we can take mesenchymal stem cells which are capable of developing into cartilage cells. So you have the scaffold, you have the stem cells here, then grow together. Once these are formed, you can give back to the implant to the patient. Using the same strategy, as we were asking the question earlier, whether for wound healing you can make even the skin cells can be regenerated. So this is actually showing the bioartificial skin, and this is normal skin. Hardly you can see much difference. Same way, even people have tried now developing muscular prosthesis, nerve guidance channels, and now many more, even trachea, which has gone to the clinic. I will show you subsequently. So using the same tissue engineering approach, so first time in Japan, Dr. Sasai's group, he is no more unfortunately, he had developed using induced pluripotent stem cell retinal cup-like cells. I will not go much more deeper into this, just keep in mind, with over a period, using a specific scaffold material like hydrogels. So they have grown these cells which are derived from the induced pluripotent stem cells, and this has formed like a retinal cup-like structure, and which is showing specific markers of the retina. So these now they are trying, in fact, the clinical setting. They have seen in a couple of patients also there are some improvement in their vision, which is wonderful, in fact, to know. Also been developed by Jürgen Noblik. So he was the first one to develop in vitro brain-like organoids, because these are miniaturized cells, not brain. Our brain is quite big. So miniature scale, even they have optic cup-like structure. So these also have been developed using the same strategy as you can see here. So using bioreactor, so they have expanded the cells and specific scaffold material. Using that, they have developed specific miniaturized organs which have shown the characteristics of brain cells. Another group also, same thing in Ohio, they have shown optic stalk, cerebral hemisphere-like structures. So even they are seeing a cephalic picture and beginning of an eye. So these are possible using the newer strategy and technology that has come into being now. One can think of utilizing the stem cells even to generate miniaturized organ and understand the development of organ per se. And this is what I was telling about the trachea, which has gone into the clinic. There are two successive patients have been treated and they are doing quite well. And their further follow-up studies also have shown the beneficial effect of this tissue engineering trachea or the windpipe. So now I will show some of the work that we have done using bone tissue engineering we are doing in the lab. So we have used, because bone is majorly the ceramic part and there is also specific polymer like collagen. So using those two, we have shown those as bioactive. They form the mineral scaffold. Mineralization they have shown. Even the same thing here I am not showing. Using the mesenchymal stem cells which are capable to form osteocytes. So using the ceramic polymer scaffold or even the metallic scaffold, what we have shown, the mesenchymal stem cells in fact are differentiating into osteocytes and the bone-like cells. They secrete also osteocalcin, that is the mineral component of bone, and showing the differentiation. So this is what, just to show or bring to the light that what are the possibilities that stem cells can give us. How best we can utilize the stem cells in understanding not only the development, even utilizing that in a clinical setting. So we are doing also in vivo experiments with animals, and we are showing also incorporation of these and bone, like if there is any bone defect, so whether it is replenishing or not, that work is going on in the lab currently. The another thing which is very exciting, I'm sure all of you would appreciate, not only having the scaffold material and then incorporating the stem cells and growing those, now people are saying whether we can use customized one using the decellularized matrix. So the meaning of decellularization, if you are getting rid of all the cells, so there will not be any immunological problems. But the scaffold, the skeleton will remain. Then you inject the cells, like here, you have the heart. So using specific detergent, especially SDS people use, they have shown also the success with that. Then you are decellularizing this. So your skeleton is intact and you know what cell can go where. Then try to inject the specific cells at that particular place. Then you can get the restoration of the particular organ. And exactly to resemble also the size. So you don't have to like miniaturized organs that I was telling about organoids. So here you can get the organs as such. So in this case, after decellularization is done. So this is a group they have published in fact in 2014, earlier in 2008. So using rat and human cadaveric tissues, this from the rat tissue and this is from the cadaveric one, lung and heart. So they have done the decellularization, then recellularization by injecting iPSC derived or embryonic stem cell derived cardiomyocytes. As you can see here, just once, yeah. See, after decellularization, so decellularization, whatever you have seen here, the same thing have been incorporated with cells and it is showing pulsating activity. So it is acting like the normal heart again. And they have got all the signature of the ECM components and the cardiac muscle specific expression, gene expression. So this shows even another way you can decellularize and recellularize. It's not only confined to animal tissue. Now many groups are trying even using the plant. So the next slide would show. So this is actually nothing but a spinach leaf. So if you take the spinach, like you can see this looking like the veins and arteries. So this also the spinach leaf has been decellularized. Now people have tried even different plant materials actually. So after decellularization, they have injected the iPSC derived or MSC derived cells there and they have triggered the system to behave like cardiomyocytes. This is actually cardiomyocyte cluster, which I cannot show the video. That's somehow I could not get that. But what it shows the fascinating approach, how people are utilizing the interdisciplinary knowledge and different, even interspecies. The plant wants decellularization and recellularizing with cells of animal cells and then getting the specific tissues of animal origin. And this, what is the advantage of that? Because here you are not getting that much immunological complications, no ethical concern. Then you are getting the cells of your interest. And if it is forming a tissue like, then one can understand how the tissue development is taking place. Then you can use specific factors to block specific signaling or activate specific signaling, one can do a number of downstream studies as basic biologists, which we are interested in doing. And now the present era is the era of tissue engineering. So not only using the strategy that I discussed, one can print the organ of interest. So for that, what one requires, one has to first image the organ that one is interested, either X-ray, CT scan, MRI. Then you have to have the CAD or the computerized approach, how exactly you have to design. So you have to design using specific strategies. Then you have to select the materials that will hold or make the skeleton, either synthetic polymer, natural polymer, or extracellular matrix, which actually help in cell-cell adhesion. Then you select the cells, whether you are taking the mature cells or pluripotent stem cells, multipotent stem cells. Then once you have all this in picture, you have to combine both, just like inkjet printer, or there are now micro extrusion or laser assisted approach. You can print layer by layer. So this is what the layer by layer printing is done. Then you are getting the entire thing. It has to be cultured further and maturation has to be done. Then you will get the entire organ in the picture by layer 3D printing or 3D tissue printing, bioprinting. Same actually in the German scientists, they have done, which I showed earlier also, using the 3D printing technology. So what they have done, they have actually taken even the same decellularized kind of structure, and exactly they have printed at the same places where it can scan that system, and after printing, you can get the exact replica of the tissue or the organ that you are interested in. This is also the same thing, and this can be utilized for various applications, drug screening, in vitro disease model, cell replacement therapy, and obviously by tissue engineering, we are getting the entire organ in picture. And another ambitious proposition, as I was telling also in the pig heart, what they have done, the transplantation, whether you can inject the stem cells in between, halfway through gestation, and when the pups are born, they will have some, these will be chimeric. So they will have specific organs which will have some human cells. So, but this is something very ambitious, but ethically people might not accept it, but technically it is possible. In fact, in Japan, this particular group, Hiromshu, he has got the permission to make the humanized organs using pig. Even they have demonstrated in rat. So pancreatic cells, in fact, they are developing. So when the pig, because from the patient, the cells will, iPS cells will be taken, those will be injected into the pig blastocyst. And once the piglets are born, their pancreas will have the human cells. So whether that can be transplanted. But these are actually the technologically feasible. But when it comes to clinic, the feasibility one has to study at length before bringing to the clinic. But now Japan government has given permission for this. The same thing as I was telling about the rat model, I'll just give this. Not only that, now it comes even making designer babies. This is ethically incorrect, but in human cases, actually, it may not be possible. But the technology is there, and in the animal kingdom, it may be possible. So when you are having the cells, any cells you are bringing to the iPS stage, you can develop the gonads, the primordial germ cells. So if it is female or male, depending on that, female cells are transplanted to the ovaries, male cells will be transplanted to testes, and then eggs and sperm, those can be fused and it can be implanted, and you will get the embryos. Now what I was telling about the I think the next slide, yeah, these are the pups born using the same approach. So this is just to show the cartoon, like you went for a simple blood test and got cloned by mistake. So the thing that I was telling, I think the next, yeah, this is the slide that I was talking about. So if you see, these are the two very, very recent studies. So using the transient reprogramming, they have reversed the age by 30 years. So this is another approach the aging researchers, they are trying to do, whether because of an age-related disorder, whether we can reverse that using the same approach of rejuvenation or rewiring the system. So this has already been demonstrated by this group, Rex group, and last week's paper using 3 small molecules like TTNPV, that is It's actually activating the retinoic acid receptor the pathway. And there is another azakenpolon, that is another mimic of this KLF1, which is one of the Yamanaka factors. It inhibits differentiation, activate Wnt signaling. Another molecule WS1 that inhibits NF-kappa B, so the differentiation is inhibited. So using small molecules, they have reprogrammed the system to a totipotent state. So in that case, you don't even require any fusion to make a zygote. So if you have any cells, you are making induced pluripotent cells, then you are bringing to the even the totipotent level where you will have the extra embryonic placental cells generated, embryonic proper the ES cells are generated. So in that case, you are having all the cells, whatever are required for developing into an organism. So think about this approach. So if you are thinking in the veterinary field, you don't even require parthenogenetic means. This is kind of you are bringing any cell and bringing to the level where the totipotent cells are there. Agent when you require, you can direct the cells to become one cell versus the other. So this is the technological advancement which has come into play now. And one can easily appreciate the kind of rationale or the knowledge, know-how has gone into this field and what it can do wonders in the field of basic biology, especially in the field of development, and the paramount importance these cells carry in the field of like a therapeutic implication that these cells might have in future. So now actually I will come to the end of my talk. So whatever I have shown already, the different advancements and what are the implications these cells can be, where all you can get, how you can generate, and what are the specific implications in the field of understanding not only normal embryogenesis, mechanism of disease, even the effect of drug. Okay, I skipped that slide here. It is just the pharma industry, specific factors. Drug screening, one can also use the stem cells for. So this, and even early development, the teratology, and the implications I already discussed about. So where we stand now in terms of cell replacement therapy, these are different disorders, specific stem cells which have been utilized, especially in LV Prasad, if you see, they are doing wonderful, giving actually vision to patients using the limbal or corneal epithelial stem cells. So it's really wonderful to see that. And same also in bone disorders. Diabetes, it's still it's at its infancy, but as we know, hematological disorders, it has gone to the clinic. Many people are trying in cancers and other diseases. And Japan and other places, they also have tried using in the fetal stem cells in Parkinson's disease, even iPSC derived cells in the retinal replacement, retinal disorders. So this is where we are today. And so whatever was there earlier, we are thinking like in the kind of a fascination or the science fiction, now it has come to reality. So where one can manipulate genes and gene editing is possible now. One can grow the organs, tissue engineering, drug designing and delivery, therapeutic cloning, transgenesis in animal kingdom. These are all possible. So one can think how best one can utilize this technique or technology that is there already and to bring more newer knowledge, newer strategies to make it even much more promising for the future. Thank you all. I will just end with this slide where it says the joy of discovery is certainly the liveliest that the mind of human can ever feel. Those who don't know the torment of unknown cannot have the joy of discovery. French physiologist Claude Bernard, who has said this. And thank you all. So any questions, I am happy to answer now.
Thank you, Dr. Nivedita. The session is open for discussion. Any questions?
If anybody has any doubt, you can write to me, email me. So no problem. And I'm certainly sorry for all the disturbances in between.
Yeah, it's okay. Sometimes.
Yes.
Yes, sir.
Thank you, ma'am, for the lecture. I want to ask ma'am regarding scaffold. As you mentioned, it should be biopolymer and it should degrade as the cells grow and mature. So, how much time, efficient time, it must be present or it must sustain in the organ to make those cells support or grow?
Yes, that usually we do in vitro. We see also the release kinetics of that, and depending on the cell type that we are interested in, we have to think of how long it can reside and how soon it should degrade. So if it degrades very fast, it is not good. If it stays quite long also, it's not good. Based on that, we select the polymeric material or any kind of material for that matter. So then we try to also see in different combinations. It's actually, it depends. It's a lot of permutation combinations you need. And there is no constant kind of thing, okay, this much time you have to keep it. So we have to study all these parameters, then we decide how long we can keep that and what should be the proportion of material that we are using. And whether it resemble the in vivo scenario, then once we implant, it should slowly degrade. So that's what we keep in mind.
Thank you, ma'am.
Sure. Any other question?
Any other questions?
Sorry, I have to keep my video off because I was not very sure that how to work. So,
Anyone else?
any questions? Okay, I saw already the question was
Questions
already.
are. So thank you so much, Dr. Nivedita, for joining us today. Despite we had some
My pleasure.
glitches, it was a very good presentation. Wonderful. Thank you so much once again.
Thank you so much. Thank you. It's my pleasure. Thank you.